स्टार्ट करिए।
हाँ, मीटिंग शुरू हो गई है।
करें स्टार्ट?
हाँ सर, अब
ऋषिका
सब
जी
लाइव है, सब क्लियर है।
सर अभी
ज्वाइन
ज्वाइन हो जाने का उसको फिर करते हैं। तो सर कर देते। जी सर, थोड़ा एक बार ज्वाइन हो जाने देते हैं।
आवाज आ रही है ना यूट्यूब पे? आयुष आयुष आवाज आ रही है? हेलो आयुष, आवाज आ रही है यूट्यूब पे लाइव?
आ रही है, आ रही है।
हेलो
आवाज आ रही, आवाज आ रही है, आवाज आ रही है। आ रही है, आ रही है।
ये स्टार्ट कर दीजिए अब। ठीक है, 33-35 बंदे आ गए हैं, धीरे धीरे आते रहेंगे। हेलो हेलो,
सर ओके सर।
हाँ स्टार्ट करिए।
The scientific world. The scientific world is to be less threatening than was feared. It is to make safe for human beings, and the way to make it safe is to reflect on the foundation of knowledge. Good morning, everyone. I am Ishika, Connection Network, the profound platform that is science era. It is the platform running under the guidance of our respected chairperson, Mr. Sudhanshu Kumar Jha. It was inaugurated by Honorable Shripad Yesonik Ji, Minister of State for New and Renewable Energy, Government of India. Scientific Era is a permanent platform that supports global academic and research excellence through its system. It offers webinars for real-time knowledge sharing, host academicians and researcher awards to recognize outstanding achievements, and publishes an e-magazine to disseminate the latest research and insights. These services foster collaboration, celebrate innovation, and help connect the academic community worldwide. We have already organized two successful webinars. One in November on the topic Advances in Ayurveda Formulations, Integrated Science and Tradition. Second in January on the topic Targeting Dynamic and Instability to Combat Chemotherapy Associated Sequelae. And today we are honored to organize third webinar on the topic Nano Formulations in Pharmaceuticals. We are excited to have you all at the today's webinar. This will be an opportunity for us to learn from each other and grow together. Thank you everyone for joining us today. We will be discussing everything and giving you practical tips that you can use in your own work. We will also take questions throughout the webinar, so please feel free to type them in the chat box and ask at the end of the webinar. And now, without further ado, I would like to hand over the further key to Mr. Abrar Ahmed sir. He is the Executive Research Editor in Scientific Era. He is the Assistant Professor in Department of Pharmacy Practice at SVS Paramedical College, Sundarburni in Jammu and Kashmir, having number of research papers and patent in his name. Sir, please.
Thank you, Ishikaji, for your kind words. Good morning, everyone. Myself, Abrar Ahmed, working as an executive research editor at Scientific. And I think it's my honor and privilege to welcome you all to the third national webinar organized by Scientific on the theme, we can say, nano formulations in pharmaceuticals. And I think nanotechnology has revolutionized the pharmaceutical industry, offering groundbreaking advancements in drug delivery, target therapies, and improved bioavailability. And I think this webinar aims to explore the latest innovations in nano formulations and their impact on modern medicine. And I think today's event has been made possible through the dedication of our organizing team. Let me introduce our team, our organizing secretary, Mr. Sudhanshu Kumar Jha. And I think we are privileged to have Mr. Sudhanshu Kumar Jha as the organizing secretary of this webinar, uh, who is currently serving as a scientist research associate uh, at the All India Institute of Ayurveda, Government of India, New Delhi. And I think Mr. Jha is also the founder, chairperson, and editor in chief of the Scientific Research and Development Firm, which was officially inaugurated by the Honorable Shri Shripad Yaso Naik Ji, Minister of State for New and Renewable Energy, Government of India. Mr. Jaz has done remarkable contributions to research and innovation. He has earned a prestigious Young Researcher Award 2024. Mr. Jha has published over 35 research articles in renowned journals. And he has also contributed to 5 book chapters, authored 5 books on national and international platforms, and published a book on Amazon. Additionally, he holds 10 published patents, 3 copyrights, 8 design grants, and 2 international design patents from the United Kingdom. And I think his participation in over 70 plus national and international conferences, seminars, and workshops highlights his commitment to research, knowledge dissemination, and societal development. And I think we are truly honored to have Mr. Jha's leadership and expertise guiding scientific era towards excellence. And our new who is convener, Mr. Sunny Chauhan, who is expert resource person and a research associate at Jubilant Bioseas Limited, Noida, at Scientific Era. And I think, of course, we are privileged to have our eminent speaker, Mr. Gopal Saras, who is working as an assistant professor at Dayanand Education Society's Dayanand College of Pharmacy, Latur, Maharashtra. And I think Mr. Gopal Sir's expertise in nano formulation will provide us with valuable insights into cutting edge advancements in pharmaceutical nanotechnology. And we're excited to have you all with us today for what promises to be an informative and engaging session. Now, without further delay, let me invite Mr. Sunny Chauhan. Please proceed the session. Over to you, Mr. Sunny, sir.
Thank you, Brasa, for your such kind of words. Now I will like to thank to our Sudhanshu sir, chairperson of to provide me the opportunity for our third webinar in nano formulation in pharmaceutical. I truly appreciate your trust and support, and I am looking forward to making the best of it. Now I am like to welcome to our speaker, Mr. Gopal Kumar Lohia, sir. He is an M Pharmacy in Quality Assurance and Assistant Professor in Department of Pharmaceutical Quality Assurance at Dayanand Education Society, Dayanand College of Pharmacy, Latur, Maharashtra. He has completed his bachelor in Maharashtra College of Pharmacy in 2008 and masters in Pharmaceutical Quality Assurance in 2011 from Maharashtra. He has total 14 years of teaching experience in UG and PG level and published 25 research papers in national and international reputed journals. He has granted 3 Indian design patent, published book and chapters in it. He is like a member of Association of Pharmaceutical Teachers of India. He has given various poster and oral presentation in national and international conferences, and he has worked as a co-chairman for a social media committee at the Indian Pharmaceutical Congress 2023 held in Nagpur. He has awarded prize for poster presentation at various national and international conferences and attended 72 national and international conferences, seminars, workshop, faculty development program, continued education program, and now he is registered as a PhD scholar at Global Mansarovar University, Sehor, Bhopal. His area of interest of research is quality assurance, formulation and development, analytical method development, pharmaceutical analysis, documentation, and validation. He is actively involved in various values added educational courses, environmental and social awareness, personality development, healthcare sector, professional ethics, student-centric co-curricular and extracurricular activities. Please, sir. Gopal, sir.
Hello, sir. Thank you very much for giving my introduction. So, I'm audible?
Yes, sir.
Yes, sir. Yes, sir.
Okay. So, first of all, I am very much thankful to the chairman of the scientific era and the entire team of the scientific era for organizing this wonderful webinar. My also best wishes for the bright future of the scientific era organization, which leads the global academic and research excellence. It is actively involved in the various activities which enforces students, faculty members, research scholars progress. I wish all the best for the future of scientific era, and I am thankful to the chairperson, Mr. Sudhanshu Kumar Jha sir, chairperson, the respected chairperson of this entire organization, for taking this lead, and give my all the best for the bright future here. So now we moving towards our presentation held on, which is on the topic that nano formulations on the pharmaceuticals. So here we will start it. So now I will share the presentation. Let me know, sir. Is it visible to all of us? The slides?
Yes, sir. Yes, sir. It is.
Okay.
It is
Now
visible.
moving towards this presentation topic, which is on the nano formulations, which are used in the pharmaceuticals for the drug delivery. So myself, Mr. Gopal Lohia, I am working as an assistant professor at the Dayanand Education Societies, Dayanand College of Pharmacy, Latur, Maharashtra. So here, let's have the discussion about this topic. Nanoformulation in pharmaceutical, it refers to the use of nanotechnology to enhance the delivery and the stability and the effectiveness of the pharmaceutical drugs. By manipulating the size, shape, and the surface properties of the drug particles at the nanoscale level, usually the size is between the 1 to 100 nanometers. Whatever the nano formulations are there, that size is between the 1 to 100 nanometers. And uh, during the development of the formulation scientist can improve its pharmacokinetics. We know pharmacokinetics means what? It is the ADME, absorption, distribution, metabolism, and the excretion. Okay, and by changing it, we can change the therapeutics of the drugs, that the drug can be delivered to the blood. Now, what is actually a nano formulation? It can be defined as the formulation utilize the nanotechnology to enhance their therapeutic efficacy. They are specifically designed to improve the delivery and the performance of the existing drugs by reducing its toxicity, improving the solubility, and increasing its viability. We can see various formulations are there, like the nanoparticles are there, liposomes are there, nanotubes, micelles, nanocapsule, dendrimers. We can use it to deliver the drug at the various site. My and numerous applications are there, like for the in the treatment of cancer tumor, the drugs we can call as the anti-neoplastic, which is used in the treatment of cancer, anesthetic, analgesic, and the antibiotics. So these are the various formulations that are there. We can see, like in the form of nano emulsions, micellar formulations, dendrimers, liposome, that by using the lipid carrier like the cholesterol, the phospholipids, and the various polymeric nanoparticle like the solid lipid nanoparticles, and all of this. So by changing its method of synthesis, by doing the change in the size and the shape of that particle, okay, so we can change its property, and we can target the nano formulation at the active sites, and we can control also the drug release of our formulation. So इसके बेनिफिट्स क्या है, हम देखेंगे कि नैनो फॉर्मुलेशन के इसका यूज क्या है, कि इसके एक्चुअली हम किसके लिए यूज करते हैं. एक्चुअली द कन्वेंशनल फॉर्मुलेशंस आर इन द मार्केट. So what are the advantages or the key benefits of the nano formulation over the conventional dosage form? So the first benefits or application we can see here, that we can increase the solubility of the poorly soluble drugs. Who those are, if you see the BCS class, biopharmaceutical class of the drugs, BCS class, four classes are there, 1 2 3 4. The drugs which are comes under the class 2, class 3, class 4, having the limited solubility and the limited permeability. So for those drugs we can improve their solubility, we can improve their drug release by using, by the converting them in the nanosystems, by converting them in the nano formulations. How? That first, how we can increase the solubility? Simple. We are going to increase the surface area of that formulation. For example, nanoparticle. By increasing the surface area of the drug, we can increase the more solubility of the drug. Okay, like a second application we can see here, that targeted drug delivery. Nanoparticles can be targeted to the specific site. Like how the specific site, for example, in the treatment of tumors, various phenomena are there. By using the ligands, we can attach on the surface of the drug, and we can target to the cancer cells or the tumor cells on the receptor, cancer receptor. So the anti-cancer drug directly go to the site of the cancer cell, and they can kill the cancer cell. And in this way we can prevent the inhibition or the killing of the normal cells of our body. So this is the main advantage of the nano formulation, that we can target the drug at the specific site, that specific site of tumor cells. Okay, so we can allow the more precise drug delivery, and that will also, it will reduces the side effects and also effectiveness, and improves the effectiveness of the treatments. Then control release. By using the nanocarriers, we can control the release of the drug. So which can helps us to maintain the therapeutic efficacy, therapeutic levels of the drug in the body. That is the bioavailability for the extended period of time. So which ultimately, what is the advantage of this one? Which will reduces the need of the frequent dosing, and ultimately it will improve the patient compliance. Now next advantage of this one, we can enhance the bioavailability. What is bioavailability? That is the availability of our drug in the bloodstream. So by using the nano formulations, we can do this. How? We can overcome to the various barriers, like we are knowing the blood brain barriers or the intestinal mucosa. Okay, by converting our drug in the nano size, we can we can pass to the blood brain barrier by the size, by reducing the size up to the 5 to the 15 nanometer. For example, uh, gold nanoparticles, by limiting their size between 3 to 15 nanometers, we can pass the blood brain barriers. Now we can also improve the stability of our formulations. How? We can protect the sensitive drugs which are sensitive to the photosensitization light, which are thermolabile. We have to protect, we can protect from the heat, we can protect from the oxidations. For examples, nanoformulations like the liposomes, nanoparticles. By using this, we can encapsulate the drug. So we can protect it from the different environmental factors. So we can prevent it from their loss. Also, next use we can see here, that we can reduce the toxicity. How? By controlling the release and the targeting of the drugs, we can reduce the overall toxicity and the side effects of the medications. How? If you see the case of the cancer, so by delivering the high doses of drug directly to the cancer cells can minimize the damage to the healthy tissue. So we can do the target to the specific site of the cancer by using the different phenomenons. So we can reduce the toxicity which occurs from our drug. So we can target to only cancer cell. It will not kill our normal cells. अभी हम देखेंगे डिफरेंट एग्जांपल्स ऑफ द नैनो फॉर्मुलेशन की नैनो फॉर्मुलेशंस के एग्जांपल्स मार्केट में क्या है. मार्केट में बहुत सारे टाइप के नैनो फॉर्मुलेशंस अवेलेबल है. इसके हम ड्रग को इनकैप्सुलेट कर सकते हैं, ड्रग को हम इम्बेड कर सकते हैं. Various examples are there. The first example is a very famous example. We all knows that we are listening about the nanoparticles. Okay. Now see more examples: dendrimers, micelles, nanospheres, nanogels, polymerosome, nanocapsules we can formulate, nanosuspensions, nanoemulsions, liposomes, neosomes, exosomes, transferosomes. So various formulations are there. So we can convert our drug into this type of nano formulations. So let's see it. So first we will discuss about the nanoparticles. So as we know, nanoparticles क्या होते हैं? These are the solid particles with the range usually between the range 1 to 100 nanometer. Okay, so by using this, we can encapsulate the drug and we can control its release, and we also target the drug to the site. So they are made up of the different material like the polymers, lipids, and the metals. In the slide, you can see this is the morphological structure of the nanoparticles by using the SAME and TEM studies. SAME means scanning electron microscopy, TEM transmission electron microscopy. By using this, we can see the morphological structure of the nanoparticle, size and shape. In the diagram, you can see the size. You can see that it's shown the 20 nanometer, and various shapes are there. Spherical shapes, nanotubes are there. In the various shapes, we can observe by using that using that analytical technique, same and tame. So here we can see the different types of nanoparticles. It is usually divided into three types of nanoparticles. We can see first is a organic nanoparticles, which are okay, which comes under which various formulations we can see. Various examples of organic nanoparticles are liposomes, solid lipid nanoparticles, nanostructure lipid carrier, polymeric nanoparticles, protein-based nanoparticles, carbohydrate-based nanoparticles. Second type is the inorganic particles. Various examples we can see here: silica, titanium oxide, zinc oxide, iron oxide, gold nanoparticles are there, silver nanoparticles are there. And the third type is the carbon-based nanoparticles. Okay, in the carbon-based nanoparticles, we can see the graphene, which is extract from the graphite, carbon black, the fullerene, quantum dots. These are the various formulations under the carbon-based nanoparticles. So various shapes we can create by using the different methods. I already told you that the size is between the range 1 nanometer to the 100 nanometer. We can check the size of the nanoparticle by using the different analytical instrument like the zeta sizer, by which we can measure the particle size of the nanoformulation. And the different shape of nanoparticles occurs, that cubic shape, rod, spherical, ellipse, plate, star shaped, all that things, oval shape, triangular, star shape, flower shape, needle, worm shape. Different shapes nanoparticles are available. We can formulate it. So ultimately, what is use? इसका use क्या है? We can improve the stability of the formulation, having the good thermal conductivity. They can tolerate the high temperature. Okay, high temperature they can tolerate it. We can make the formulation stable. The mechanical strength is good, biocompatible, and the biodegradable. These are the main advantages about these nanoparticles, and they are flexible. We can functionally, they can becomes the flexible ones. So we can see the different type of applications here by using the nanoparticle. In the which areas this nanoparticle formulations can be applicable, that in the treatment of cancer therapy, in the drug delivery, we can control the drug release, in the treatment of wound healing, treatment of diabetes, gene therapy, genomics. We can encapsulate, we can incorporate the antimicrobial agents, so we can do this application. Antimicrobial application we can obtain in the field of tissue engineering, biosensors, in the cosmetic and the food division, food systems. Now we can see about the some parameters we can see about the nanoparticles. In the diagram, you can see the different aspects we have to see here, that how they are changing the colors. Three parameters are we can see in the slides here: the aspect ratio, thickness. By changing the thickness, the we can change the color, and the size is about the of the nanoshells is around the 140 nanometer, nanorods, and the gold nanoparticle. By varying the concentration of the gold, these are around the size of 50 nanometer. We can do this. So these all type of nanoparticles showed characteristics, colors, and the properties with the variation in the size and the shape. What is the use of it? It is it is utilized in the bioimaging applications. Bioimaging applications, where we have to study the images, clear images of the biological membranes, cells, tissues. So we can, by using this. So as in the figure, you can see the color of the solution changes due to the variation of the aspect ratio, the nanoshell thickness, and the percentage of gold concentration. So by we can change the colors. Okay, by doing the alteration of any of the discussed factors, that the aspect ratio, the shell thickness, and the percent gold, that influences the absorption properties of the nanoparticles, and hence the different absorption colors are observed, and that is the main application. It is used in the bioimaging. We can study the structure of the biological membranes. We can see that. So we can see, uh, I already told you that the size range of nanoparticles is in between the 1 nanometer to the 100 nanometers. So these are the different nanoparticles. We can convert it at a polymeric nanospheres. You can see in the slides, polymeric nanocapsules, polymeric micelle, liposomes, liposomes by using the lipid carriers like the cholesterol, phospholipid, soy lecithin, and all these things. Dendrimers, inorganic nanoparticles. For example, mesoporous silica nanoparticles, carbon nanotubes, iron oxide nanoparticle. It is used in the magnetic resonance imaging. Iron oxide nanoparticle are mainly used for the MRI techniques, magnetic resonance imaging, gold nanoparticles, quantum dots, and these are the different shapes of nanoparticle we can obtain: star shape, cube shape, spear shape, plate shape, and the rod shape. So we have some discussion about the various characteristic of the nanoparticles. So first characteristic we discuss here, that the size and the surface area. The size is usually from the 1 to 100 nanometer. Size कितनी होती है nanoparticles की? वैसे तो आप देख सकते हैं कि आपको 1000 नैनोमीटर तक देख सकते हैं, लेकिन जो एप्लीकेबिलिटी है that is about the 1 to 100 nanometers. So you can target your drug to the site. Okay. So by using this, they exhibit the increased surface area to the volume ratio, which can enhance the interaction with the surrounding materials or the biological tissues. इसका यूज क्या है? Smile says small size of the nanoparticles that they can penetrate to the biological barriers. They can penetrate to the cell membranes. They can penetrate to the blood-brain barrier, as I said, by reducing the size of, for example, gold nanoparticle. So we can able to cross the blood-brain barrier also. Then the surface properties. Okay, the surface properties means like the charge, hydrophobicity, various functional groups. So that can be modified. So we can improve their interaction of the nanoparticles with the biological systems or the environment. So which ultimately helps in to target the specific cells or the tissues. For example, the cancer cells. Hello, I'm audible.
Yes, sir. Yes, sir. You are audible, sir.
Yes. Okay. Yes. So by doing the surface modification, we can help in targeting the specific cells or the tissue. That is the applicable in the case of the cancer cells, or we can reduce the toxicity. Then the quantum effects. So quantum effects, that the material exhibit the unique optical, electrical, or the magnetic properties that not seen at the macro level. So at the nano level, we can see that. For example, nanoparticles, for example, gold nanoparticles or the magnetic nanoparticles, which can be applicable, which can be useful for the diagnostic purpose or the therapeutic purposes. So this is the ultimately the classification of the nanoparticles, I have told you that it is classified in the organic nanoparticles, inorganic nanoparticles, and the carbon-based nanoparticles. So in the organic nanoparticles, examples are there various liposomes, solid lipid nanoparticles are there, nanostructure lipid carriers, polymeric nanoparticles are there, protein-based nanoparticles are there, carbohydrate base. In the inorganic, silica, titanium oxide, zinc oxide, or the zirconium dioxide, iron oxide, gold, and the silver. And in the carbon-based nanoparticles, the fullerene, quantum dots, graphene, and the carbon black. Let's see some advantages of the organic nanoparticles, that they are biocompatible and the biodegradable. That is the main application of nanoparticle. Biocompatibility is most useful, that our body is compatible those to the nanoparticles. It is also biodegradable. No need to again remove it from our body. It is flexibility. By doing the surface modification, we can target to the site. By doing the changing its structure, a different attachment, we can change it the different functions. We can release the control of the drug. About the inorganic nanoparticles, they are unique electrical, magnetic, and the optical properties. Different sizes में हम obtain कर सकते हैं. Various sizes, structure, and the geometrical structure, having the good mechanical properties about the inorganic nanoparticles, and also show the catalytic activity and exceptional mechanical properties shown by the carbon-based nanoparticles. Also show the good thermal conductivity, means stable at that temperature, chemical stability, various advantage like the surface functionalization and the biocompatibility and the environmental remediations. Various example we can see here, the inorganic nanoparticles. In that, let's see some example, that magnetic inorganic nanoparticles. In that, examples are there. Iron oxide nanoparticles, mainly used in the magnetic resonance imaging. Gold nanoparticles. So we can, by reducing the size of gold nanoparticle, we can target to the brain targeting. We can deliver our drug to the brain. Silver nanoparticles. So silver nanoparticle basically used in the antimicrobial use to kill the different bacterial microorganisms. In the ceramic, silica nanoparticles, titanium oxide nanoparticles, alumina nanoparticles. In the semiconductor type, zirconium dioxide nanoparticles, zinc oxide nanoparticles, and these are the carbon-based nanoparticles. We have seen that one example. We have seen that fullerenes, graphene. It is extracted from the graphite, the carbon black, and the carbon quantum dots. So these are the all the allotrope of the carbons. Okay, these are all the allotrope of the carbon materials. So these are the various examples of the organic nanoparticles. Protein based, various proteins, example like the collagen nanoparticle, albumin nanoparticle, gelatin nanoparticles are there, casein nanoparticle, whey protein nanoparticles, and the plant protein nanoparticles. In the lipid based, liposomes, by using the lipid carriers, cholesterol, soy lecithin, and all its, we can embed the hydrophilic and the hydrophobic drug. We can release the drug. Okay. Solid lipid nanoparticles, various nanostructure lipid carriers. In the polymeric, by using the chitosan, the alginate nanoparticles, PLA, PGA nanoparticles, polyglycolic acid, okay, all that. And these are the carbohydrate base, that the starch nanoparticles, dextron nanoparticles, and the cyclodextrin nanoparticles. So let's have see about some types of the nanoparticles. In that, the first type is the metallic nanoparticles. So in the metallic nanoparticle, the first subtype is a gold nanoparticles. Okay, we can see this gold nanoparticle, which is used for the drug delivery in the diagnostic purpose, in the bioimaging, magnetic resonance imaging, and by using the biosensors. If we see some advantage of these gold particles, that I already told you that it is a biocompatible and that can be easily functionalized with the various biomolecules. Different applications are there. We can control the release of drug. We can deliver the protein or the antigen. Okay, as we know, the antigen antibody reaction is there, so we can that antigen directly elicit the antibody response. Therapeutic ligands, that on the surface of this nanoparticle we can attach the ligands, so which will target to the cancer receptor cells. So we can target to the cancer cells by using these targeting ligands. Like that, the targeting antibodies, okay, in the nucleic acid deliveries, DNA, RNA, drug delivery, microbial agent or the toxin removals, in the adjuvant targeting, optical probes imaging, tag dyes for the imaging, and the photothermal therapy. So these different applications we can came by making the gold nanoparticles. This is a very wide field. Just I am just showing you uh, what are the advantages of these nano systems, nano carriers. Second type is the silver nanoparticles. Silver nanoparticles, it is mainly used for the, it is the antimicrobial properties. They can kill the microorganisms. So that are used in the wound dressing, coatings, and the treatment of the infections. So various sizes, various shapes, you can see that one, that the cubic nature, needle, worm shape, rod shape, oval shape, triangular, star shapes. Okay, so these different shapes we can obtain of this silver nanoparticles, spherical shapes and star shape and the cubic scales and the needle-like structure, the square structure. So we can obtain. You can see this is the morphological structure of the silver nanoparticles. We can see that one, that the size is less than the 100 nanometer. Okay, different sizes. We can, nano means that is the range, broad range, 1 to 1000 nanometer. So 20 nanometer, 100 nanometer, 500 nanometers, 200 nanometers, and all that things you can see here. These are the again the types of the nanoparticle. That is the iron oxide nanoparticles. It is mainly used for the MRI purpose, that is a magnetic resonance imaging purpose. Okay, so that is a magnetic targeting by using this iron oxide nanoparticle. So this is the morphological structure of this iron oxide nanoparticle. Different shapes you can see here, different needle-like structures, cubic structures, square structures. Okay, we can form, we can formulate the iron oxide nanoparticle. Its main use, इसका main use क्या है? For the bioimaging, for the magnetic resonance imaging. These are the morphological structure of the iron oxide nanoparticle. This is the SEM or the TM photographs, transmission electron microscopy, and the scanning electron microscopy. So we can see the different iron oxide nanoparticle in the different shapes. This is the polymeric nanoparticles, which are made from the polymers. Which are the polymers? Like a PLGA. PLGA means polylactic co-glycolic acid, polycaprolactone, or the chitosan, or the chitosan. Okay. So what is the use of these polymeric nanoparticles? It is used for the sustain and the control release of the drugs. So by using this, we can encapsulate the hydrophobic drugs. You can see this one, different polymeric nanoparticles we are used different. By using this, we can formulate There are different nano formulations like the nano capsule, nanospheres. If you see the structure, you will come to know the drug is encapsulated inside. In the outer core, the polymeric core is there. Polymeric membrane is there, which is made up of the PLGA, polylactic co-glycolic acid, or the polycaprolactone, or the chitosans. Okay. It is mainly useful to encapsulate about the hydrophobic drugs. Next, we can see about the dendrimers. Dendrimers, dendrimer, if you see that these are the nano size, radially symmetric molecules with well-defined, homogeneous, and monodispersed structure, and that is typically a symmetric core and the inner shell. Symmetric core at the inner shell. Okay, so due to this structure, these are used to encapsulate the drugs or the other therapeutic agents. So here we can control the drug release, also the targeted delivery to the specific cells or the tissues. Okay, so this is the dendrimers, different types of the nano formulations. This is a lipid-based nanoparticles. In that, the example is this is a liposomes. By using the हम इसमें क्या करेंगे, लिपिड कैरियर्स यूज करेंगे ड्रग को रिलीज करने के लिए. ओके, सो इसमें हम हमारा ड्रग है इनकैप्सुलेट कर सकते हैं. कौन सा ड्रग कर सकते हैं? हाइड्रोफिलिक भी कर सकते हैं, हाइड्रोफोबिक भी कर सकते हैं. By using the lipid carrier like the cholesterol, soy lecithin, by using the different the lipid carriers. So we can see if you see the liposomes, you will come to know that in the center is a hydrophilic core is there. There is a hydrophobic bilayer. Okay, this is the cholesterol molecules are there. These are the phospholipids. If you see the more detailed one, various applications of these liposomes, that by using the liposomes you can increase the drug solubility for the poorly soluble drugs, the drugs which are comes under the BCS class 3 or 4, that is having the low permeability and the low absorptions. So in that we can improve their increase their permeability, increase their absorption, so ultimately increase their bioavailability. So these are the conventional liposomes. By the इसमें हम हाइड्रोफोबिक ड्रग इनकॉर्पोरेट कर सकते हैं. ओके, इसमें सेकंड एग्जांपल है कि हम लिगैंड यूज करके टारगेटेड कर सकते हैं जहां पे हमको हमारा ड्रग रिलीज करना है, फॉर एग्जांपल एट द कैंसर साइट और द मे बी इन द केस ऑफ वैक्सीन्स और द different things. So ligand targeted liposomes are there. These are the polyglycolated liposomes are there by using the PEG, polyethylene glycols. So this is the lipid based nanoparticles are there. So this is the one of the method commonly used for the preparation of liposome. लिपोसोम बनाने के लिए एक मेथड है, इसका नाम है थिन फिल्म हाइड्रेशन मेथड. तो इसमें क्या यूज होता है? बाय यूजिंग दिस रोटरी इवेपोरेटर. Okay, we have to add our active moiety, active drug, active hydrophobic drug, the organic solutions. We have to dissolve in it by using the rotary evaporator. By applying the vacuum, we can obtain the thin lipid film on the bottom of that rotary vacuum evaporator. After stirring that and mixing and storage, there is a formation of multilamellar liposomes. So हम multilamellar liposome को फिर क्या करेंगे? नैनो फिल्टर से पास करेंगे. नैनो फिल्टर साइजेस फिल्टर्स आर अवेलेबल. तो इसमें हम पास करने के बाद by using the extruder we can obtain the unilamellar nanoliposomes. Okay, so we can deliver the various types of drugs which may be the hydrophilic, may be the hydrophobic, or may be the amphiphilic. This is one of the method I am showing there, that this is the laboratory method. By using the rotary vacuum evaporator we can prepare the liposomes. So this is about the solid lipid nanoparticles. These nanoparticles are composed of the solid lipids and is used for the control and the sustained release of the drugs. So the outer layer is made up of the surfactant, different surfactant like the non-ionic surfactant, solid lipid, and there is an active API. Our drug is incorporated here. Solid lipid nanoparticle. Next is a nanostructure lipid carriers. Means they are in the combination of the solid as well as the liquid lipids. So they, if we compare with the solid lipid nanoparticle, they show the greater stability, also show the good drug loading capacity. So this is liquid lipid core, drug is there, and this is the phospholipid bilayer. This is the surfactant. Usually the non-ionic surfactants are there. Drug is there, solid lipid material. Okay. So these are the nanostructure lipid carriers. Various example we can see here. So ultimately, what is the mechanism? How the nanoparticle is going to be act? So that is the main principle involved here. That is the ROS means what? Reactive oxygen species. Okay. So by the mechanism is the oxidative stress. Oxidative stress, it means it is an imbalance between the free radicals and the antioxidants. So by how? By doing the DNA damage to the our cancerous cell, by doing the protein denaturation, mitochondrial dysfunction, lipid peroxidation, DNA adapt gene mutation by doing the gene mutation, protein oxidation, enzyme dysfunctioning, oxidative DNA damage, and the cell membrane damage, we can apply the nanoparticle to the target site and we can obtain the drug released at the specific site. For example, the tumor site or the cancer site. So various application we can see that by using the organic, inorganic, and the carbon-based nanoparticles, that it is useful in the drug delivery, in the cancer treatment, in the antimicrobial activity, and all that ones. Next, we have to discuss here that is about the quantum dots. Quantum dots means what? क्या है? These are the fluorescent colloidal nanocrystals. Colloidal means what? Having the very smaller size nanocrystals. Smaller size means the size range is between the 2 to 10 nanometer. इसमें क्या होता है? इसमें होता है inorganic core होता है, which is responsible for the optical properties, and it is having the semiconductor shape. So with the lining size 1 to 10 nanometer in the diameter, it is usually made up of cadmium selenide, cadmium telluride, indium phosphide, and the indium arsenide. Okay, so these are the semiconductor shells. For example, these are the semiconductor materials. So that can be synthesized by the colloidal methods or the electrochemical methods. So what is the main use of these quantum dots? The size you can see here, 2 to 50 nanometers are there. It is used in the bioshooting, where these particles serve as a contrast agents by using the bioluminescence, fluoroluminescence. Okay, they serve as a contrast agent, giving the much better resolution than the existing fluorescent colors. So this is the use for the diagnostic purpose, a diagnostic use. That is for the in vitro and the in vivo detection and the analysis of the many biomolecules. In vitro means the studies which are conducted outside the body, and in vivo that drugs is injected to the animals, okay, or the human beings. In vitro containing what? Various artificial models, and in vivo that drug is directly we are going to administer to the animal or the human being. This is again nanocrystals, various types of nanocrystal we can formulate, that coated nanocrystals, liposomes, Nano suspensions we can made in the tablet, hydrogels, microneedles, microparticles. So that can be applicable in the different route of administration, that oral route tablets, nano suspension, for the parenteral purpose, the liposomes, all that. So the what is the size range of the nanocrystals? That they wide range is there, that is a 10 to 800 nanometer. So drug nanocrystals, it is consisting of the pure drug in that range. These are the promising approach for enhancing the drug delivery by improving the solubility, dissolution, bioavailability for the drugs whose solubility is lower. Various market formulation we can see here. I have quoted two nano formulation example. That one formulation is tricor phenofibrate. It is a nanocrystal formulation developed by the Abbott Laboratories for the improve the oral bioavailability of the drug phenofibrate. And the second nano formulation is the triglide phenofibrate, another formulation developed by the Sky Pharma and improves their oral bioavailability. So these are the structures. Let's have some various nanosystems, nanocarriers like the neosomes. This is no doubt this is similar to the liposome. Some slight changes are there. That in that the use of the non-ionic surfactant is there. No, the lipid carriers usually the cholesterols are there. Okay, so these are the nanocarriers. These are used to okay. So these are the nanocarrier that are used to deliver the drugs to the specific sites of the media. And that I have told you that it is made up of the non-ionic surfactants, और ये कोलेस्ट्रॉल और non-ionic surfactants से बनाए जाते हैं. But if you compare between the liposome and neosome, they are less expensive when you compare to the liposomes. So by using the neosomes, you can again encapsulate the hydrophobic drugs as well as the hydrophilic drugs, and you can target the release of the drugs. Okay, so you can see the neosomes, the ligand you can incorporate on the surface of neosomes, so that will go to the receptor, and we can obtain this phenomenon. This phenomenon is known as what? This is the receptor mediated endocytosis. So ligand is bind to the receptor, cancer receptor, and the endocytosis. Endocytosis means what? The neosome engulf the cancerous cells. As we know, the simple principle involved in the complexometric titration, ligand and the metal ions. Ligand क्या करता है? They donate the electron pair to the metal ion, और metal क्या करता है? Electron pair accept करता है. So this is the complexometry principle. By doing this, by obtaining by absorption of the ligand on the surface of the neosomes, we can target the drug at the cancerous sites. Ethosomes, similar formulations are there, but some slight changes are there. Ethosomes, you can see ethosomes. These are the soft malleable vesicles composed mainly of phospholipids, ethanol, having some higher concentration in the water. So this is usually applicable for the transdermal drug delivery. Okay, so we can pass our drug through the skin membranes. So ethosomes are usually this. So this is for the to deliver the drug to the skin. These are the transferosomes. This is also applicable that to the transdermal drug delivery. So these are the self aggregates with an ultra flexible membrane, so we can deliver our drug to the at the site to the skins. So these are the vesicles which can carry the drugs across the skin. तो इसमें क्या होता है, इसमें होता है एज एक्टिवेटर्स होता है, इथेनॉल कॉन्सेंट्रेशन है, फॉस्फोलिपिड बायलेयर्स, एंड वी कैन अगेन इसका यूज है कि हम हाइड्रोफिलिक मतलब वाटर लविंग और हाइड्रोफोबिक या लिपिड लविंग ऐसे दोनों ड्रग हम इसमें ट्रांसफर कर सकते हैं। ये आपने सुना होगा नैनो रोबोट्स, इसका मेन हम देखेंगे नैनो रोबोट्स क्या है। नैनो रोबोट्स, दीज आर द टाइनी मशीन्स, छोटे छोटे मशीन्स है, so that can be used for the medical diagnosis and the treatment. So by using this we can deliver the drugs, genes and the other substance to the specific sites in the body. For example, cancer cell detection, cancer biomarker detection, chemodynamic therapy, sonodynamic therapy, phototherapy and the immunotherapy. So we can see these are the different micro and the nano robots used in the medicine. By using this, we can deliver the pharmaceuticals, various biological hormones, genes, living cells, inorganic therapeutic substances. It is also used in the surgery, biopsy और सैम्पलिंग. अगर डॉक्टर को कैंसर सेल कैंसरस है कि नहीं चेक करना है तो डॉक्टर क्या करेगा, दे एडवाइस अस दैट डू द बायोप्सी. बायोप्सी मतलब क्या, वो हमारा सेल का सैंपल लेते हैं और दे विल चेक इट दैट वो सेल्स कैंसरस है या नॉन कैंसरस है. टिश्यू पेनिट्रेशन, इंट्रासेल्युलर डिलीवरी, बायोफिल्म डिग्रेडेशन in the diagnosis purpose, and it is also used in the imaging purpose, जहाँ में रेडियोलॉजिकल इमेजिंग होती है, ऑप्टिकल हो, अल्ट्रासाउंड हो, मैग्नेटिक हो, रेडियो बेस्ड हो, तो इसका इसमें यूज है. तो इसमें कैंसर में क्या यूज है इसका, नैनो रोबोट्स कैन डिलीवर द एंटी कैंसर ड्रग्स टू द डिसीज सेल्स विदाउट हार्मिंग द हेल्दी सेल्स. क्योंकि नैनो का वही मैकेनिज्म है कि हम हेल्दी सेल्स को कोई हार्म नहीं करेंगे, जो कैंसरस सेल है उसको हम टारगेट करेंगे. इसका और एक एप्लीकेशन है कि वुंड हीलिंग. So nanorobots can speed up the healing process by releasing the biodegradable mesh into the wound. Biodegradable, फिर से आपको वो निकालना नहीं है. Automatically it will be absorbed in the biological membranes. So this is again application in the tissue regeneration and the medical imaging techniques. So in the nanotechnology, nano formulations, various natural product based natural biopolymers are there that we can use in the nanotechnology. For example, we can see that various natural biopolymers that can obtain from the plants, like the example starch, cellulose है, गुअर गम है, गम अरेबिक है, या फिर इसमें पॉलीमर से जो एनिमल से ऑप्टेन करते हैं, काइटिन, काइटोसन, okay, glycoso aminoglycans, hyaluronic acid obtained from the marine sources, chitosan. Okay, various polymers we can obtain from the microorganisms by doing the fermentation process. Various polymers like the dextran, gelen gum, xanthan gum, and the bacterial cellulose. Various polymers we can obtain from the algae, there is the alginates, galactans, and the carrageenans. So various we can see the natural compounds which we can extract from the plants. Phytoconstituents are there, so that can be used as a nanomedicines. There is example you can see which is having the antimicrobial action, caffeic acid, the coumarin, the eugenol, menthol, various neuroprotective phytoconstituents, curcumin, anti-cancer drugs are there. Okay, vincristine, vinblastine, and the resveratrol, silmarin. Okay, different antioxidants are there. Gingerol, glycyrrhizine, cyanidine, curcumin, and various anti-inflammatory natural compounds are there. The colchicine, capsine, quercetin, and the epigallocatechin 3-gallate. So this can be used in the nanotechnology. So these are the, you can see the different natural phytoconstituents are there which are encapsulated by using the different nano formulations. That you can see द नैनो सस्पेंशन, माइक्रो इमल्शन है, नैनो लिपोसोम है, इसमें माइसेलर फॉर्मेशन है, नैनो कैप्सूल्स है और सॉलिड लिपिड नैनो पार्टिकल्स है। ओके, सो इसमें हम क्या कर सकते हैं, हम इसमें क्या करेंगे, जो भी फाइटो कॉन्स्टिट्यूएंट है वो इसमें इनकॉर्पोरेट करेंगे, ऑल दिस थिंग्स। ओके, इसमें जो भी हमारा नेचुरल प्रोडक्ट्स है, जो ड्रग एक्टिव मोइटी, दैट वी कैन इनकैप्सुलेट, और हम डिफरेंट डिफरेंट रूट्स ऑफ एडमिनिस्ट्रेशन में इसको अप्लाई कर सकते हैं, ओरली हो, इंट्रावेनस हो, नेजल हो, इनहेलेशन हो, ट्रांसडर्मल हो, ऑफ्थेल्मिक। ओके, तो इन दिस नेचुरल बाय प्रोडक्ट्स हम इसमें यूज कर सकते हैं, डिफरेंट डिफरेंट। Okay, so these are some of the nano formulations for the phytoconstituents or the phytocompounds that we have seen. Let's have see this liposome, इसमें क्या होता है, फॉस्फोलिपिड बाइलेयर होता है। नियोसोम्स में क्या होता है, यूजिंग नॉन आयोनिक सर्फैक्टेंट्स। फाइटोसोम्स में क्या है, फॉस्फोलिपिड ड्रग कॉम्प्लेक्स है, इसमें फाइटोकॉन्स्टेंट यूज करते हैं। ये माइसेलर्स फॉर्मुलेशन है, पॉलीमेरिक नैनो पार्टिकल्स, गोल्ड नैनो पार्टिकल्स, मेसोस्पोरस नैनो पार्टिकल्स, ग्राफीन नैनो पार्टिकल्स हम इसमें से एक्सट्रैक्ट करते हैं, ग्राफाइट से करते हैं। सिलिका नैनो पार्टिकल्स और एक नैनो स्ट्रक्चर लिपिड कैरियर है, सॉलिड लिपिड नैनो पार्टिकल। ओके, एंड ऑल दैट थिंग्स वर नैनो इमल्शन, नैनो सस्पेंशन। सो इसमें जो फैक्टर्स है वो कौन से जरूरी है, जरूरी फैक्टर्स है, वी कैन वेरी इट, साइज हो, शेप है, उस पर सरफेस चार्ज है, रिस्पॉन्सिव स्टिमुला है, हम डिफरेंट डिफरेंट रिस्पॉन्सिव स्टिमुला यूज कर सकते हैं। टेम्परेचर इंड्यूस हो, ओके, प्रेशर इंड्यूस हो, पॉलीमर बेस हो, ओके, दैट बाय the doing the surface modification we can change the activity of that phytoconstituent by converting it into the nano formulations. So अब ये फाइटोकॉन्स्टिट्यूएंट्स हम किसमें यूज कर सकते हैं, एंटी कैंसर, एंटी इंफ्लेमेशन, एंटी बैक्टीरियल, एंटीऑक्सीडेंट, एंटी न्यूरो डिजनरेशन, phototherapy and the imaging. So these are the wide range of this one. So ultimately nano formulation जो भी है, nano carriers है, nano systems है, nano medicines है, इसका यूज किसमें है, हम देख सकते हैं कि हम we can enhance the solubility, we can enhance the absorption, so ultimately we can enhance the bioavailability, enhance the permeability of our drug, we can increase the stability of our formulation, हम easily हमारा ड्रग क्या कर सकते हैं, इनकैप्सुलेट कर सकते हैं, अंदर हम इम्बेड कर सकते हैं, हम टारगेट कर सकते हैं एट द स्पेसिफिक साइट्स। सो हमारे नॉर्मल जो सेल है वो किल नहीं होंगे, तो हमारी जो इम्यून सिस्टम है वो स्टेबल रहेगी। ओके, सो दैट इज अ reduce the immune recognition, it is also the reduce the medicine degradation, increase the solubility, हम इसमें टारगेटेड ड्रग डिलीवरी सिस्टम अप्लाई कर सकते हैं, इजी इन द एब्सॉर्प्शन है, शेल्फ लाइफ हम बढ़ा सकते हैं, हमारे फॉर्मुलेशन की हम शेल्फ लाइफ हम बढ़ा सकते हैं। अगर हम कंपेयर करें कन्वेंशनल ड्रग डिलीवरी सिस्टम, फॉर एग्जांपल डिफरेंट डिफरेंट जो आपको पता है, ओरल सॉलिड, ओरल में सॉलिड है, लिक्विड है, सेमी सॉलिड है, गैसेस है, जैसे कि सेमी सॉलिड में ऑइंटमेंट है, क्रीम्स है, इमल्शंस है, ओरल सॉलिड में पाउडर्स है, ग्रैन्यूल्स है, टैबलेट कैप्सूल है, लिक्विड में सिरप सॉल्यूशन है। तो इसमें क्या प्रॉब्लम है, दैट ड्यू टू द फर्स्ट पास इफेक्ट, ड्यू टू द जीआईटी एंजाइम्स, हमारा ड्रग क्या होता है, वहां पे डिकम्पोज होता है, डिग्रेड होता है। So this orally consumption of this all this formulation like the tablet, capsules or the syrup that can be damaged by the enzymatic reaction and the lower pH environment. Lower pH क्या होता है, हमारा स्टमक का pH क्या होता है, एसिडिक होता है। ओके, अभी इसमें हमने क्या किया, इंप्रूव किया, हमने कंट्रोल ड्रग डिलीवरी यूज की, हाइड्रोजेल, इरोडेबल मटेरियल, पॉलीमर मैट्रिक्स, डिग्रेडेबल मटेरियल। तो इसमें क्या है, this system is fronting as a drug aggregation due to large size of matrices, delay in the release time, less soluble, less target, ऐसे कुछ डिसएडवांटेज है इसके, कंट्रोल लिस्ट के। तो उसको ओवरकम करने के लिए हमने क्या किया, यहाँ पे नैनो मेडिसिन, नैनो फॉर्मुलेशंस डेवलप किए, जो हमने आज देखे, डिफरेंट नैनो पार्टिकल्स, फॉर एग्जांपल गोल्ड नैनो पार्टिकल हो, सिल्वर हो, आयरन ऑक्साइड हो, सॉलिड लिपिड नैनो पार्टिकल्स है, नैनो फाइबर्स है, नैनो ट्यूब्स है, नैनो स्पीयर्स है, डेंड्रीमर्स है, नियोसोम्स है, कार्बन बेस नैनो ट्यूब्स है। तो इसका यूज क्या है, दैट इज अ टारगेट स्पेसिफिक, ट्रांसपोर्ट द ड्रग टू द इफेक्टेड एरियाज विदाउट डैमेजिंग द हेल्दी सेल। ये बहुत जरूरी है कि आजकल आपने देखा कि कैंसर का जो भी कैंसर के पेशेंट बढ़ रहे हैं, उनको ट्रीटमेंट है मार्केट में, रेडियोलॉजिकल ट्रीटमेंट है, उनको कीमोथेरेपी है, लेकिन उससे क्या हो रहा है, नॉर्मल सेल भी मर रहे, कैंसर सेल के समय नॉर्मल सेल भी मर रहे। तो हमारी क्या हो रही है, जो इम्यून सिस्टम वो कोलैप्स हो रही है। तो उसको हमने प्रिवेंट करने के लिए हमने क्या किया, नैनो फॉर्मुलेशन हम डेवलप कर रहे हैं, नैनो मेडिसिन वी आर गोइंग टू डेवलप है। इसमें अगर हम एग्जाम्पल आपको बताया, तो इसमें अभी कैंसर के लिए कोई एग्जाम्पल है, जैसे हमने डॉक्सोरुपिजिन के लिपोसोम्स डेवलप किए जो कि ओवरियन कैंसर के लिए है, ओलापेरिब के लिपोसोम्स है जो कि द ट्रीटमेंट ऑफ द ओवरियन कैंसर, ओलापेरिब। सो ऐसे डिफरेंट डिफरेंट नैनो फॉर्मुलेशंस हम अभी डेवलप कर रहे हैं, जो कि we can compare if this conventional control is, so more advantages we are getting in the nanomedicines. So these are the ultimate the application of in the drug development. We can see that these nano formulation allow the drugs to be delivered directly to the cancer cells, potentially improving the effectiveness of the chemotherapy. Okay, as we know, कीमोथेरेपी मतलब क्या, हम हमारे एंटी कैंसर सेल्स, हमारे एंटी कैंसर ड्रग्स हम देते हैं। ओके, तो वी कैन रिड्यूसिंग द साइड इफेक्ट्स। हम ये देखते हैं कैंसर पेशेंट को साइड इफेक्ट होते हैं, वो हम अवॉइड कर सकते हैं। वैक्सीन, okay, by using these nano systems we can deliver the vaccines, we can improve the immune response, so we can provide the better protection against the different diseases. In the genomics, in the gene therapy, so these nano system nanoparticles can be used as the vehicle vectors, so to deliver the genetic material, that's the DNA or the RNA, that to target the cells for the gene therapy applications. Again, the protein and the peptide delivery, that help to protect the fragile proteins and the peptides and the enabling their use in the drug delivery, स्पेशली जो बायोलॉजिकल से बायोसिमिलर्स है, तो हम ये उसमें प्रोटीन और पेप्टाइड ड्रग डिलीवरी कर सकते हैं। सो अल्टीमेटली कंक्लूजन क्या है, आज के जो पीपीटी का कंक्लूजन क्या है, सो दैट्स द नैनो फॉर्मुलेशन होल्ड द great potential for the revolutionary drug delivery by improving the pharmacokinetics, by changing the absorption, distribution, metabolism, and the excretion. We can improve the bioavailability and the targeted delivery of the pharmaceuticals drugs, active pharmaceutical ingredients, active moieties, maybe the active moiety. Various example, nose to brain drug delivery. By using the nose, we can deliver the drug to the nasal system. We can pass our drug to blood brain barrier. We can also avoid the blood brain barrier by passing the drug through the trigeminal pathway or the different pathways. So what are the challenges? There are still challenges related to the safety, manufacturing, and the regulatory approval that need to be addressed for the widespread adoption. So अभी भी इसमें कुछ challenges है जो हमको overcome करने हैं, stability of the formulations, all that things. So we have to overcome this. So this is about all this nano formulation जो कि अभी market में available है और अभी use हो रहा है. So lastly, I am thank you to the chairperson of the scientific era, Mr. Sudhanshu Kumar Jha sir,
Thank you, sir.
for
Thank you, sir.
inviting me for as a speaker for this webinar. So thank you very much, sir.
It's
Thank you,
been,
sir.
sir, thank you so much. It's been our honor that as you are not, you are just my senior and my good friend, and I am happy that you accepted our invitation. and the way you just express your knowledge about nano formulation. I'm really very happy today because हमारा पहला वेबिनार यूट्यूब पे सक्सेसफुल रहा और उसको आपने ही सर एक तरीके से प्रोसीड किया। तो साइंटिस्टिक एरा और मेरी टीम की तरफ से आई एम वेरी thanks to you, and we are very grateful, and we look
Thank
forward
you
for
very
more
much.
opportunities together, sir. And I
Sure,
would
sir,
like
sure.
to thank to my team as well, their dedication, your dedication, sir. And finally, जो मैं चाहता था कि हमारा YouTube पे proceed हो तो आज हुआ.
Yes,
So
yes.
thank you so much, sir, and the team.
Thank you very much.
And due to some busy schedule of mine, I won't be able to join today. So thank you so much, sir. Abraj ji,
Yes, yes.
continue.
Thank you, sir. Thank you. Anyone having questions related to this nano formulations can ask.
Yes.
Anyone having questions can ask openly.
चैट बॉक्स में कुछ होंगे क्वेरीज तो आप देख सकते
You can drop your questions in the chat box,
हैं चैट बॉक्स.
and the questions will be answered by Mr. Gopal, sir.
Sir, I just wanted to ask one thing. हमारा अभी यहाँ मिनिस्ट्री में फॉर्मुलेशंस पे तो काम चल रहा है, but we want some like phytochemicals and plants that are suitable for cancer as a nano formulation. So if you have some work, or if you already you have expertise of over like decade plus, तो अगर आप हमें उसमें थोड़ा हेल्प करें तो we can make a like एक हम प्रोजेक्ट डाल सकते हैं गवर्नमेंट में, या फिर हम उस पर कुछ आर्टिकल वगैरह एक अच्छी जगह पब्लिश कर सकते हैं।
चलेगा. श्योर
तो
श्योर.
कुछ अगर सर फाइटोकेमिकल्स या कुछ प्लांट ओरिएंटेड हो जिसको हम नैनो फॉर्मुलेशन कैरियर्स में इनकॉर्पोरेट करके कैंसर के लिए या
यस.
सीबीडी डायबिटीज के लिए कर सकते हैं, तो सर आप कुछ बताएं, या फिर कुछ हमारा कुछ ऐसा प्लान हो तो वी कैन
चलेगा.
लाइक डिस्कस ऑन दैट।
श्योर सर, हम उस पर स्टडी करेंगे. श्योर हो. इसमें बहुत वाइड रेंज है स्टडी करने के लिए.
यस
बहुत
सर,
सारे फाइटो कॉन्स्टिट्यूएंट है, क्वारसिटीन
यस
वगैरह.
सर,
क्वारसिटीन के लिपोसोम्स है अभी, उसमें
यस
हम
सर।
बहुत सारा स्टडी कर सकते हैं.
ओके सर।
वेरियस.
हम्म। तो किसी का कुछ है क्वेश्चन वगैरह राज जी?
Yes, sir. YouTube के चैट बॉक्स में सर क्वेश्चंस है कुछ.
ओके ओके ओके।
गोपाल सर, आप देख रहे हो सर YouTube पे, YouTube के चैट बॉक्स में कुछ क्वेश्चंस है आपके लिए.
एक्चुअली मैंने यूट्यूब पे लिंक ओपन नहीं की है. कुछ होंगे तो मुझे आप बोलिए. आई विल टेक दैट. कुछ
I
होंगे
think
तो आप भी बोलिए.
there is one question, sir. He has written marketed product is in market. कुछ नैनो फॉर्मुलेशंस का कुछ मार्केट में है ऐसा कुछ प्रोडक्ट.
है ना सर, बहुत सारे हैं. अभी जैसे ओलापेरिब है, मैंने बताया कि ओलापेरिब के नैनो पार्ट उसके लिपोसोम्स है. ओलापेरिब जो एंटी कैंसर ड्रग है, उसका यूज होता है इन द ट्रीटमेंट ऑफ ओवरियन कैंसर के लिए. उसके बाद डॉक्सोरुबिसिन है, उसका भी मार्केट में लिपोसोमल फॉर्मुलेशंस अवेलेबल है. तो ऐसे बहुत सारे फॉर्मुलेशन है जो हम आईवी रूट से वगैरह देते हैं. इस पे टू मच फॉर्मुलेशंस आर देयर, दे आर present in the market. I can see some more example. Danorubicin है, doxorubicin है, और डिफरेंट एग्जांपल्स है. इसमें बहुत सारे हैं सर ऑलरेडी मार्केट में. ऐसा नहीं है कि मार्केट में नहीं है. उसका रूट आईवी रूट से वगैरह हम अभी यूज में है वो, और उस पे बहुत सारा रिसर्च गोइंग ऑन है.
Okay, thank you, sir. One more question is there, sir. Is there any good nano injection possible over there? Nano injections are possible or not?
नैनो इंजेक्शंस है ना भाई, वही तो मैं जो बताया आपको. नैनो लिपोसोम्स मतलब लिपोसोम्स में क्या करते हैं, साइज जो है वो रिड्यूस करते हैं, और उनको हम जैसे कि डॉक्सोरुबिसिन है, ओलापेरिब है, इनको इंट्रावेनस रूट, सबक्यूटेनियस रूट, हम इसको नैनो साइज में इंजेक्शन थ्रू ही देते हैं. वो वर्ड सिर्फ अलग उनको ध्यान में नहीं आया होगा, क्योंकि हम उसकी साइज रिड्यूस करके हम जो टारगेट साइट है जहाँ पे कैंसर सेल प्रेजेंट है तो वहां पे हम उसको टारगेट कर रहे हैं.
One more question is there, sir. Do enzyme responsive nanoparticles play in biomedical applications?
ओके.
Yes.
तो एंजाइम्स भी है ना, जैसे हम एंजाइम्स में देखते हैं कि लॉक एंड की मॉडल्स है, एंजाइम मेडिएटेड रिस्पॉन्स है. तो हम जैसे एंजाइम मेडिएटेड कि जैसे आपका जो एंटी कैंसर मॉइटी है या फिर एक्टिव मॉइटी है वहां पे जाके आपके आपके रिसेप्टर पे बाइंड होगा और वो आपका ड्रग वहां पे रिलीज होगा. तो एंजाइम मेडिएटेड ड्रग डिलीवरी है इसमें. दिस इज वन ऑफ द एप्लीकेबिलिटी इन दिस है सर इसमें.
Last question is there, sir. Is it possible to formulate nano formulation having Vinca drug?
विनक्रिस्टिन विनब्लास्टिन. यस यस. और जैसे फाइटोकेमिकल्स है ना, विनक्रिस्टिन विनब्लास्टिन जो जिनका यूज है, जिनका थेरापेटिक यूज है एंटी कैंसर, प्लांट से एक्सट्रैक्ट होता है, उसकी बहुत कम कम क्वांटिटी की कॉस्ट बहुत ज्यादा है. तो उसके भी फॉर्मुलेशंस डेवलप हो रहे हैं. उस पे भी सर्च चल रहा है सर. कि मैंने जो इसके
Okay.
लिए स्पेसिफिक बताया कि फाइटोकेमिकल जो मैंने बताया एक स्लाइड में कि फाइटो कॉन्स्टिट्यूएंट्स मैक्सिमम यूज करके उसका भी हम कन्वर्जन कर रहे हैं नैनो फॉर्मुलेशन में. ड्रग रिलीज कर सकते हैं, कंट्रोल रिलीज कर सकते हैं.
Okay, thank you, sir. I think these are the questions that have been asked by the members over there. And I think
Okay.
as we come to the conclusion of this insightful and engaging session, I take this opportunity uh, to express my heartfelt gratitude to all those who made this webinar a success. And first and foremost, a special thank you, sir, for your valuable time and for sharing your expertise on nanoformulation as pharmaceuticals. And I think your insights have deepened our understanding of this field, and we truly appreciate your contributions. And obviously, I would also like to extend my sincere appreciation to our organizing secretary, Mr. Sudhanshu Kumarja, for his leadership and vision in making this webinar possible. And also want to thank you, our convener, Mr. Sunny Chauhan, for his continuous support and contributions. And of course, uh, it has been my privilege to serve as the coordinator and host for this event. And I think to all the attendees, your participation, curiosity, and enthusiasm have been truly commendable. Your engagement in today's discussion highlights the importance of such knowledge sharing platforms. And before closing the session, I think I would like you to all the members who are present over there. Uh, Certificates will be shared within a week, maybe in the WhatsApp group or maybe through mail. And stay
Uh,
connected with scientific era.
Abrar ji, just have to share that कुछ issue की वजह से group has been, I think, something is wrong. So if possible, we can mail you. So check करते रहिएगा अपना email ID भी, क्या पता आपको certificate वहाँ पे मिले, because it's almost like 400 registrations were there.
Yeah, within
And
a week, it will be shared with
within
you.
a week or 10 days, you will get to all your certificates. So
And one last thing, just stay connected with scientific era for future webinars, conferences, and research activities. And we look forward to bringing you more opportunities to learn and grow. And with that, we officially conclude today's webinar. Thank you all once again, and have a
and
great
again, a
day ahead.
special thanks to Gopal
Yes,
sir for their valuable
special
time
thanks
and
to
knowledge. Yeah,
Thank you sir.
and our
Thank you so
technical
much.
team and our coordinators, Abrar ji, Shika ji, Sunny ji.
thank you so much.
Well done, good job, and look forward for more opportunities like this. Thank you so much.